Witam wszystkich serdecznie na drugim dniu Maria Myśli. To jest kolejny wykład w ramach serii wykładów podczas tego tygodnia Maria Myśli. Hello, welcome everyone on the event Maria Myśli. This is the second lecture today, and I would like to introduce the professor. First in Polish, po polsku. Profesor Lars Kai Hansen jest kierownikiem selekcji systemów poznawczych na Uniwersytecie Technicznym w Danii. Jego badania koncentrują się na uczeniu maszynowym z zastosowaniem w biomedycynie, mediach cyfrowych i systemach kognitywnych. Opublikował ponad 300 artykułów na te tematy. Jego wkład w naukę obejmuje wprowadzenie i analizę zespołów sieci neuronowych, pierwsze zastosowanie predykcyjnego uczenia maszynowego do czytania w myślach w PET, koncepcja komponentów poznawczych. Jego ostatnia praca dotyczy monitorowania uwagi w czasie rzeczywistym za pomocą EEG. Na tygodniu mózgu usłyszymy od niego o Machine Learning for Brain State Monitoring based on EEG. W tym wykładzie dowiemy się o tym, jak elektroencefalografia to szeroko stosowana nieinwazyjna technologia monitorowania stanu mózgu. Sygnał EEG jest złożony i uwikłany w artefakty, stąd modelowanie i podejmowanie decyzji w oparciu o EEG stanowi poważne wyzwanie. Profesor Lars Kai Hansen dokona przeglądu postępów w uczeniu maszynowym do analizy i zastosowania EEG. Now in English, Professor Lars Kai Hansen is head of the section of Cognitive Science System at Technical University in Denmark. His research focuses on machine learning with applications in biomedicine, digital media, and cognitive systems. He has published more than 300 papers on these subjects. The major contributions include introduction and analysis of neural network assemblies, the first application of predictive machine learning for mind reading in PET and fMRI, the concept of cognitive components, and the cure for variance inflation. His recent work concerns real-time attention monitoring using EEG. At the Brain Week, Maria Myśli will hear from him about machine learning for brain state monitoring based on EEG. And about this lecture, electroencephalography is widely used non-invasive technology for brain state monitoring. The EEG signal is complex and confounded by artifacts, hence modeling and decision-making based on EEG is a serious challenge. Professor Lars Kai Hansen will review progress in machine learning for EEG analysis and applications. Po wykładzie zapraszam do zadawania pytań. Można zadawać je też w czasie wykładu na YouTubie oraz na warsztacie. So I would like to introduce you to ask questions during and after the lecture on the YouTube chat. And the professor, the floor is yours.
Thank you so much, and thank you for the introduction and for this opportunity to discuss machine learning in EEG and how we can combine the two to do brain state monitoring. What I'm going to present this afternoon is kind of an open invitation also to collaborations here. As you'll see, it's a very broad spectrum of applications that we have in mind. And the same with the tools. It's not like one single tool for one application. It's a whole suite of different ways that we are approaching the signal. And there's so many challenges and so many interesting possibilities now that we are very happy to collaborate on these issues. So in this presentation, I'm going to say a few words about the signal first, and then share my vision, how we can approach EEG with AI and machine learning tools, and in this way, hopefully connect with real-life events. So much of what we know about the brain has been obtained, this information has been obtained from laboratory experiments or laboratory equipment and so on, but we're really trying to move this outside the lab and see if we can connect with real life, and I'll try to be more clear about what that means later. And then in the second half, I'm going to discuss two very specific applications. So one is this, what we call EEG in the classroom. There's a picture at the lower right side of how that experiment was carried out, and this was published some years ago. And then finally, the sort of more recent real-time experiment we're doing with attention monitoring and control that came out last year. So let me move on to just say a few words about the signal. So EEG is interesting in many ways as a brain signal because it has no delay. So compared to the two sort of main modalities that we study the brain by, neuroimaging, functional MRI and positron emission tomography, that both are based on blood flow, meaning that the sluggish blood flow delays the perception of signals outside in scanners and so on. EEG, quite the opposite, it's instantaneous. So what we measure with the grid, as you see in the picture, upper right side, as a sort of an indication of an experiment. What you measure is what happens instantaneously in the brain. So now what we can pick up with sort of typical surface electrodes would be signals generated in the superficial part of the brain. So this means the cortex. And of course, there's now a complication that we only see part of the brain, but what we see is already a big part, an important part. The cortex is an important part of our decision making and so on. But it's also the fact that also a confounder is present, namely anything else electric in the head and in the surroundings. So muscle tension in the face muscles, eye blinking, eye motion, so on. All of these signals will generate strong confounders, sometimes of energy higher than the signal we're looking for. So these confounding signals can be so strong that they sort of, so to speak, jam the signal, the brain signal. And for this reason, we need strong inference methods. So that's why AI is supposed to be a tool for us that we can learn what to discard and what to analyze. So in this presentation, I'm going to focus on some aspects of my work that has to do with what happens at, you could say, the system level. I'm going to talk about sort of time and space constraints and also what we can mind read. So this would be the classroom experiment or the attention control. I'm not going to speak so much about how we can localize these signals in the brain. There will be a little discussion about that in connection with the smartphone brain scanner. But most of that work I'll leave for another time. It's a super interesting problem and where AI is also really helpful. So here is sort of the general vision of how we can use AI to connect lab experience with real life events. So imagine that you have EEG acquisitions tools from the complex lab equipment that you see in the upper left corner to things that are basically possible to wear in everyday life. So this is possible. So we have the mid-level systems, what we call wearable EEG. So most of the experiments that I'm going to talk about today are created with wearable EEG. So this means that we have some kind of a cap with electrodes, but then signals are transmitted wirelessly to a receiver, a smartphone, for example. But sort of the ultimate solution would be to have acquisition of EEG that is completely invisible. So one possibility is the so-called ear EEG that is developed by a colleague of mine here in Denmark also, by Kildmose. And there the signal is acquired inside the ear canal, and this means that it's basically possible to wear the signals day and night for longer periods of time. And there's another device closely related to this, but slightly more invasive, that is implanted under the skin. It's produced by a company, Unique, also here in Denmark, that also allows very long time acquisition in the wild, so basically in everyday life. Now, to connect these different modalities of EEG acquisition, we want to use the machine learning tools. So we want to create strong priors that we can learn from conventional lab-based equipment and then transfer those as priors that will help us to make inference in the more noisy and more shallow experiments that have maybe fewer electrodes and fewer measurements. So that's the big vision of the work. So one way that we use these strong priors is to use neural networks that are fine-trained to help us to analyze. So here's a very brief example of such a piece of work where we do sleep decoding. So the signal is EEG acquired while you sleep, and it's one of the main ways that EEG is actually used in medicine today, is to understand the quality of the sleep. And typically this would be patients that have problems with sleeping, and now we want to understand where the problems arise from and if there's things we can do to mitigate it. So the idea is to understand sleep through the electrical signals that different brain states under sleep are propagated to the scalp electrodes. And the classification problem that ensures, so this is trying to understand exactly at every given time what is the sleep state that we're in, including to be actually awake or to have different types of deep sleep or rapid eye movement sleep, that is the dream states. And by using visual networks that have been pre-trained on very general images and then fine-tuning them on EEG data with labels, we can use this setup that we just talked about, that you have sort of more general experience that you apply to a very specific context. And of course, more details are available in the paper. Another very important dimension to this piece of work is that we have then analyzed the network after the training, and we can see that the features that they're looking for in the EEG signal are similar to what current day doctors are looking for when they analyze sleep signals. So let's go to talk about these more wearable technologies that will eventually allow us to do neurotechnology in the wild. So this device that I'm going to show you some examples from are the two that you see in the pictures here. So in the left side, we have the unique device. So that's the implantable electrode. So it's sitting under the skin. So it's a relatively simple operation. Can be done with simple means in outpatient conditions. And the other system that I'm going to show examples from is the EEG. That is basically like an earplug that picks up a signal inside the ear canal. So here's some data from the implantable electrodes. It's a single patient we're following, and we are looking at data from more than a month of contiguous acquisition. So the patient has the device implanted, and then there's a week that where the implant is stabilizing itself, and then we start to acquire data day and night. So in the plot, so I hope you can see my cursor. So I have a plot that says from 0 to 24. So these are hours in the day, and then I have on the horizontal axis I have days of the experiments, and you can see that it's more than 30 days that we have acquired data. The blue areas are places where the signal, for one reason or the other, is missing, typically because there's a headpiece, sort of like an earpiece that picks up the signal that can fail to deliver the signal. But most of the time during the many days, the signal is acquired. In this plot, I'm showing the result of the analysis of short windows on what we call spectral microstates. So we are looking at different types of signals, and we are simply acquiring these signals over time in 1 second intervals. So there's a lot of these 1 second intervals, of course, in 24 hours and even more so in 30 days. And now we simply ask, how predictable is the signal? And try to learn something about EEG from that predictability. So we look at how easy it is to compress the signal that through the Fano inequality that gives us a bound on the predictability. So that's an upper bound, and a lower bound can of course be found simply by making a classification algorithm, a predictor. So we have both upper bound and lower bounds, and they're actually quite close here. So the predictability is sort of in that interval between the blue and the red curve. So there's a narrow interval of predictability. So now you can look at this in different ways. So the first plot is over hours for during the day. And you can see that the predictability is highest at night and slightly lower at daytime, and in the evening gets high again, so the full day. So now if we look at the total picture of the different days, there's sort of like a yellowish beginning of the signal. And we looked at the sort of indications of where the patient were. So there's a smartphone involved also that picks up the signal, but also looks at where the patient is, and we can see that this patient was actually on holidays the first week. And it turns out that in holidays, the predictability is lower. So in a way, the mind or the brain is more unpredictable during leisure. So in general days, it's more predictable at night, less predictable during day, and on the days that you're on holidays, you're even less predictable. So I think that's already by itself sort of an interesting finding from this type of very long range EEG experiments. Here's a case where we looked at the EEG. So again, this is something you can do in the wild. You can wear this device more or less freely as a experimental subject. So here we are looking at the information link between what happens in the brain and what happens in the ear canal. So is it actually so that we can measure the real EEG signals in the ear canal? That's a question. And it turns out that there's a very strong mutual information between the two. So that's the content of the paper, that we show that electrodes on the scalp, of course, the closer to the ear, the more predictable they are from the ear, but in general, they are quite highly correlated. So those signals that we acquire in the ear are actually a good proxy for what happens in most of the scalp electrodes, and in the sense, most of the brain. So this again establishes a strong prior for EEG decoding, that we can use all the information that we have acquired in the lab on the conventional electrodes, caps, and so on, and try to use that to understand what happens in those data from the ear canal that we then can acquire both at night and day and so on in patients and in more sort of classic type experiments. Let me go on to talk about the system that we developed, that we call the smartphone brain scanner, which is a way of democratizing the whole business of EEG so that it becomes easier to use. So this is a brain imaging device that we aim to build that can be used more or less freely also with a wireless cap. So let me first explain sort of what the problem we are solving is. So in the systems I've been talking about up to now, either it's been CAP or EEG and so on, measures on the surface. But as I said in the beginning, the signal arises from sources in the cortex. And of course, it would be super nice to be able where those sources were, to be able to solve the so-called source localization problem. So looking at it from a mathematical point of view, there's one really encouraging fact, and that is that this transformation from the cortical surface to the surface electrode is linear. So to a very good approximation, we can treat this as a linear mixing problem. So the many different sources on the cortical surface conspire to make these readings that we have on the surface in a linear manner. So this means that we can write it as a linear system, where the x's would be the sources in the brain and the y's would be the measurements that we have on the surface. So now, this being a linear system, we could sort of naively think that why don't we just invert the matrix? So there's a matrix relation between the two. Why don't we just invert that matrix? Now, in most cases, that's not possible because we have a relatively small number of electrodes. So for example, in the ear, we would have only maybe a handful of electrodes. And in a typical cap, we would have maybe up to 100 or so electrodes. And the brain, of course, if we want to say something relevant for the brain, we have to use many more sort of local areas that we want to speak about, maybe in the thousands and so on. So this is, in general, a quite difficult problem. And this is why we need strong priors, again, from machine learning or from other neuroscience facts that we can bring in to make this a solvable inverse problem. And in the smartphone brain scanner, we're using what we call a Bayesian inference scheme. So we solve this in short time intervals, and we develop a Bayesian inference scheme to estimate the noise so that we can be more sort of conservative in the signals when we have high noise levels, noisy conditions, and be more free if we have low noise conditions. So that takes place in real time also. So we have a high frame rate of solving the inverse problems, and then a slightly slower rate by which we update this Bayesian inference scheme. So every 10 seconds, we produce a new model to create the forward model that we can use for the inverse solution. So why is this significant in general? So of course, there are many ways that we could use this kind of 3D imaging result that we get from the smartphone brain scanner. So one obvious way would be to do sort of what we call online visual quality control. So we can simply look at the signals and see that they look sensible. The other, and that's in the papers also, is that we can use these signals to create what we call neurofeedback applications. We would say, let's pick up signals from specific brain areas and then use those to feed back to the user, and then they could either enhance signals there or not, depending on what we want to use the neurofeedback for. For training, for example, concentration or attention and so on, we can concentrate better if we get neurofeedback on how well we concentrate and so on. We can train that aspect. And that's all discussed in the papers on the smartphone brain scanner. And finally, we can improve all this by connecting it with general neuroscience. So we have these many generations of neuroscience experiments that have taught us a lot about what happens in the brain. And they are all in a 3D coordinate system describing the brain. So if we want to connect with those and understand what goes on in the brain at a specific time, we can connect that with this kind of ontological information about the brain with the 3D pictures that we get from the brain scanner. So here's a picture of how it looks in real life, and there's a YouTube link that you can go and check out where you can see it in live presentation. So basically you wear the cap, and there's a wireless transmissions device that transmits the signal to the smartphone, and then the smartphone renders the brain on the display, and you can see how the brain signals change over time, as indicated in this picture. So now there's not only sort of a possibility of looking at your brain in real life and do that at a reasonably high speed. So we have about 40 frames per second in the system. But you can also start now to look at how interesting are the signals that you pick up. So we can make small experiments, for example, brain-computer interfacing type experiments. We can do different types of stimulus presentations, or we can do neurofeedback, as I mentioned. And this particular experiment that I'm going to show you a trace of here is one where we do precisely this idea of connecting with neuroscience information. So in atlases of the brain, we have regions. So for example, we have the region of a hand motion. So the hand area, that region is relatively well defined. So we can simply ask now, let the brain scanner invert the signal from the surface electrodes to regions and ask how much signal is present in that area. And now the experiment is very similar to brain-computer interfacing. So we'll let the subject imagine to move either left or right hand, not actually moving anything, but just imagining to move the left and right hand. And we know that if the brain starts to imagine moving a hand, then there's a resting state signal called the mu wave that is desynchronized. So it basically disappears. So in this plot, you can see at time 0, when we give the cue, we have a relatively high power of the signal. So this is this resting state signal. And then as we start to imagine moving, in this case, the right hand, then the right hand area activation of the mu signal will start to decay. So now here we compare two signals. One is what we call emotives. That's the brain cap that we're looking at with the real-time system, and the biosimi system is a reference system that is sort of the top-end EEG lab type equipment. And you can see the red curve acquired with the brain scan and the blue curve acquired with the top of the line system are very similar. And they have approximately the same error rate if you want to predict whether the subject is actually now preparing to move right hand or not. So this is a strong indication that an aggregate, say like the signal acquired in a whole area, like the right hand area, for example, would be a valid indicator, even if you acquire it in the wild with a wearable device, as we have here. But we have, of course, many more ideas what you can use this equipment for. And now I'm sort of going to mention another dimension to this, and that is the fact that, of course, smartphones are abundant, they are all over the place, and the caps are very inexpensive to produce. So this means that, and this is why we called it the democratization of EEG in the first place, is that we can apply these technologies in areas where neuroscience is really a problem. So in lower middle income countries where it may even be that neither the equipment is present nor abundant expertise is present, that we can help those areas to diagnose, for example, sleep disorder, as I already talked about, or the other important area where EEG is used for diagnosis, namely epilepsy. So this is joint work with Farah Mateen at the Massachusetts General Hospital, Harvard Medical School, who's interested in bringing these technologies to the different areas of the world where this is hard to get at simply. So to make this more feasible, we have created a startup called Brain Capture that is producing the very inexpensive cabs and using this technology to help diagnose in these places in the world. But because the device is relatively inexpensive, we can also produce it in great amounts. So here's an experiment where we have 10 of these equipments in a classroom. And we have 10 volunteers, 10 students that are participating in this experiment. So this is an experiment that has been done quite a few times in other places, both using other technologies like fMRI, but also EEG. And the question is now, with this low-cost equipment and the wireless devices and so on, can we reproduce the experiment that has been done previously in a lab? And can we do it in real time? So can we monitor these signals in real time in the classroom, potentially using it, for example, for attention monitoring? So this paper from 14 is kind of proof of concept. So the paper has a number of innovations. So first of all, we show that it's possible to synchronize the acquisition in a large number of subjects in a classroom. The stimulus is a video, so you get sort of an idea. In the left side, there's a picture of sort of a black and white picture. So it's an old Hitchcock movie. A boy that is walking around in an environment, and he, by an accident, he gets a loaded gun in his hand. So he's pointing a gun at different people, and of course this is super engaging. So you're really engaged with the movie, and we can see that this engagement produces a synchronized signal in the brains of the viewers. So this is well known. So there's this previous work by Lucas Parra and co-workers that is the second reference in the list from 14. And in our work, we could not only reproduce this experiment with the low-cost wearable equipment, we could also do a very accurate description of the similarity between the traces of the signals that we get from our synchronized subjects that are actually in real time looking at the video with the experiment that Lucas did that was asynchronous. So there's one viewer at a time in a lab, right? So it's quite remarkable. So you see it in the plots in the middle below. The red curve is the response in the study over many repeated experiments in a lab. And the black curve is the study that we did here in the classroom. So this is the joint attention signal that we find synchronized among the subjects and very carefully tracing out what happens in the movie. So there's a control experiment where we chop the movie up in other bytes. So it comes in different order, permuted order. And this has much less engagement, so we can really monitor the effect of the engagement in the movie, also in real time in the classroom, which is super interesting for us, of course, that we can apply this device in the classroom, and we can hope that it can support learning and better teaching practices and so on. The final experiment that I want to talk a little bit about is an experiment that is also done with wearable device. It's not the device I just showed you. It's a device that is wearable, but is using a technique called tri-electrode acquisition. So this means that it's not only wearable, but it's also completely non-invasive. So when you just put it on, and then you have contact with the skin that makes it possible for you to acquire data basically anywhere you want to do it. So this is a slightly more costly equipment, but again, it's interesting to see whether these devices can be used in sort of more general conditions. So this experiment is another of our experiments where we want to understand whether we can take lab type experiments from fMRI and then move it to this kind of environment. So the experiment takes place over 3 days. And it's involving a type of stimulus that you see in the upper left corner. So it's a stimulus that is a mixture of a face image and a scene, in this case an indoor scene, so basically a living room. And the subject is told to either focus on one or the other and solve a discrimination task in either the face images, male, female classification, or in the scene, it's either an indoor or outdoor scene, and you have to do that classification. And now we present that to a classification algorithm, try to do mind reading to guess whether the subject is doing one or the other task. And by the predictability, we can again monitor the attention level. And it turns out that from this experiment by Bettencourt and so on, to a Brown team that did the experiment originally in fMRI, that by manipulating the mixture, so we can make it harder or easier to spot to make the classification, the visual classification, and we can manipulate that in real time according to the readout that we do. So in this sense, we can monitor the level of attention and we can manipulate it to keep a certain level. So if the subject is sort of decreasing attention, we can make the task a little harder, meaning that they will increase attention. Or if it's following very closely what is going on, then we can make it a little easier so they relax a little bit. And in this sense, we can sort of monitor and control the attention. And this is possible to do in real time. We can not only decode, we can also control. And in the experiment, they show that by this training, you actually have a positive impact on your ability to concentrate or focus attention. This was not possible in our experiment. We don't know exactly why. So it is possible to control attention, but whether we can train to obtain better attention, it's unknown. It could be that our sample was too small, or the signals in EEG simply are weaker than the ones that we have in fMRI. But the basic attention control is possible. So again, just like we did for the sleep scoring experiment, here we can also ask if we can classify the attention level in the brain. Then what is it that the classifier is picking up? So this is an important thing for all machine learning, I would say. And of course, the more complicated and more complex the experiments become, the more important is it that we can sort of open up these black box classifiers. In this case, it's a support vector machine that is doing the classification. And we have a way to do that that combines two measures. So first, we can measure the sensitivity of the classification performance to each location in the input. In this case, an image that has time versus the different channels in the electrode locations. So that's the sensitivity we can measure. But then we would also like to understand the uncertainty of that measure. And that we do by a piece of technology that we call the NPAIR. So it's a very elaborate scheme to resample the data. So we split it up in many subsets, two halves, and then we run the classifier and the interpretation algorithm on both of these halves. Because they are sampled with replacements, we have the two visualizations should be identical. If they are not, it's because of the uncertainty. And this means that we can use that resampling scheme to estimate the variance of the visualization. So not only can we show you sort of how important are the different times, different locations on the brain, on the surface of the skull, I should have said. We can measure the importance in time and also locations on the skull. And we can also give you the uncertainty of that estimate. And here it's actually what we could call C-scores that we report. So this is the mean effect divided by the standard deviation of this resampling exercise. And you can see that there are some areas that are more important than others. So for example, in the visual areas, so these are these O electrodes, they are at around 300 milliseconds after presentation of stimulus. That's an important pickup of signals. And that's of course well known that 300 milliseconds is an important time scale for this type of information gathering in the visual domain. The final thing that I want to make a small announcement of is a tool for analyzing that is also based on the same ideas that we use very strong priors to make a representation of the EEG signal. And then in this way, we can do biomarkers and so on with some confidence because we removed quite a bit of the uncertainty in the EEG signal. So this specific toolbox is looking for microstates. So these are scalp images that are stationary for some time, maybe 100 milliseconds, 200 milliseconds. So we simply do clustering of the scalp images and then find these stable clusters defined as microstates, and then the signal that we pick up is how they change over time. And in the experiments that we're doing right now, this is for biomarkers in psychiatry to early stage different types of neurodegenerative diseases, or as in particular case that we're looking at is for schizophrenia, early signs of schizophrenia, and trying to subtype schizophrenia patients. So in this domain, there's a lack of tools, and we have produced a toolbox that matches the EEG lab. It's a plugin for EEG lab, a very widely used toolbox. EEG lab is one of the dominant tools, and now it's possible to do microstate analysis also with that. There's a detailed toolbox guide if you're interested. And of course, if you're interested in collaborating on this, we are very happy to talk to you about that also. So let me reach my conclusions here, that I hope through these examples I've shown you that EEG is an interesting signal. It's neuroscience in the sense that we measure what happens in the brain, but it's also in the wild in the sense that we can bring it to completely new areas that haven't been possible before. For example, the classroom, with obvious implications in societal implications for applications. It's a very interesting signal, but also a very problematic one. It has tons of challenges. We have these ill-posed inverse problems to understand where the signal derives from, and we have the strong confounders that often have more energy than the relatively weak brain signals. And for this reason, we need to be careful when we do the algorithm. So we have to use state-of-the-art machine learning methods to use all the data in the way we can, using very strong priors. And the priors can come from other experiments, or it can come from neuroscience in general. And in this way, we can reduce the uncertainty, reduce the noise, and with the combination of EEG and AI, we can sort of claim that we can connect cognitive neuroscience and brain state monitoring, and hopefully for a better understanding in real brains, in real conditions. All of this work has not been possible without a lot of funding and very generous support from the organizations that I mentioned in the box. Much of what we have done in neuroscience has been funded by the US National Institute of Health through the Human Brain Project, by the Danish foundations Lundbeck and Novo Nordisk, and also the work that we have on the two real-life devices, the EEG and the unique device was funded by the Innovation Foundation Denmark. So all of this we're super grateful for, and it's just a lot of it we try to give back also to the community. So many of the tools that I've talked about, the toolbox for microstates, the attention monitoring and so on, are open source, and the whole smartphone brain scanner system was open source also. So it's hopefully relatively easy to use and apply in other experiments also. So with these words, let me just thank you all for your attention, and let me take your questions if there are questions.
Thank you very much. Yes, there are a few questions, but first I would like to encourage other people to ask questions because we still have some time. So firstly, do you think that such EEG measurements and sensors like Empatica could be used in the clinical setting for diagnosing seizures, detecting epilepsy, or other abnormal oscillations?
Absolutely. So yes, so I would say that the CAP-based systems are known to be used in that context, and we have validated that. That's one of the experiments that I showed you earlier, that we pick up signals that are very similar to what you pick up with more sort of conventional CAP-based systems. So the CAPs, definitely. Now, if we talk about the unique device and the EEG, this is a lot of the validation that is happening at the moment. So they lose a bit because they have a narrow field of use. Sometimes we call these keyhole devices, in the sense that they are like watching something through a keyhole, so you only see it sort of indirectly. And this is why we need the strong priors to understand what we're looking for. But the strong suit for these devices is that they can measure over a very long time. So some of these disturbances, for example, when we're looking for a diagnosing of epileptic disturbances and so on, can be quite rare. So therefore, you would like to look at the subject not just one night, for example, but maybe like we did in the experiment for 30 nights in a row to pick up these relatively infrequent events. So I think that the combined toolbox that has the CAP for more sort of full-scale definition of what the brain conditions that particular subject has, plus the wearable devices that then can measure and find these rare events over long time scales, is a super interesting combination. But of course, the future will show exactly how this pans out.
Okay, thank you. So we were speaking about the future, and the next question is, what future do you see for EEG technology, especially in the context of the EEG outside of the laboratory and further integration with AI?
Yeah, so thank you for that question. Of course, this is very much the topic here, that that we have this vision that using AI, we can indeed use these signals, although they are heavily confounded by being in real life. So we have data that has been acquired while people are driving, for example, and we all know that there's a very strong noise signal, electric noise signal in a car and so on. So clearly, some of these signals, they will be so heavily impacted by noise signals that we cannot use them. But we have really interesting algorithms also for separating the signals, so we can separate noise signals from brain signals, and in this sense, hopefully, we can make some use even of these signals in hard conditions like driving or bicycling or running and so on.
All right, thank you. And the next question is, let me open. Yep. How do you interpret synchronization of the oscillations between subjects? Is it connected with the content of the presented stimuli, or can it be modulated by the personality of the subject, their tendency to submission, or more passive or active way of processing the cognitive stimuli?
Yes, that's a very good question. So we actually discussed that in the paper. So I encourage you to take a look at the paper on the classroom EEG. So we show that so first of all, the driver for this signal is indeed the changes in the video. So every time the picture is changing, there's a response in the visual cortices. And the strength of that signal depends on attention. That's sort of the general model. And we can show that by showing the video in a condition where there's very little attention. Excuse me. So it's possible to do sort of an intervention to actually demonstrate that effect, that attention is modulating the response. But as you say, the response itself is a response to the stimulus. So if there was no video, then we wouldn't see that synchronization.
Should I ask another question? Yeah. All right. So this one. Do you think there are some areas where EEG, in the context of presented research, could fail?
Absolutely. So EEG is a super interesting experiment, and it has these degrees of freedom that we can take them outside the lab and so on, which is harder for other technologies like fMRI and positive emission tomography and so on. But for example, the technology involved in, or the physiological signal that we pick up by the positive emission tomography, for example, is very well understood. So in that sense, that signal is better understood than the EEG signal. We're still learning a lot about what the EEG signal is, the sources, and how they react to different types of activity and so on. So much is unknown about the EEG signal. It may fail. It may be that we simply, for some subjects or for some conditions, we simply cannot see the important dimensions. EEG is much stronger cortical, so we are losing some of the signals that arrive from more central brain areas, and they can be super important for some diseases also. So there are also limitations, definitely physical and sort of how our understanding has progressed in EEG that will limit the what we can use it for. Yes, a good question.
All right. So let me ask the last question, please. Do you think that if we were all to have a permanent EEG sensor at all times, may the data acquired from it be abused by commercial entities for targeted advertising? Will it become a privacy concern in the future?
Yes, that's another super nice question, and thank you for this question. So we actually wrote about that also. We have a paper where we very detailed sort of lay out all the difficulties in having access to neurotype experiments. So if you let's imagine a simple scenario where you acquire EEG data, because now it's really accessible and so on. It's relatively inexpensive, so you maybe simply want to buy a device and play with it. Would be tempting to publish your data, right? So put them on Facebook or something. Now, if you post a picture from a Friday night party on Facebook, you know that you'll be laughed at the Monday morning, right? You can sort of understand what the impact of revealing that picture, what that will be. That's not the case with EEG signals. We don't even know what they can be used for. We don't even know how detailed you can decode them and so on. So it may be that tomorrow there's a technology that can decode something from your EEG data that was not aware about today. So EEG signals and heart signals and other type of physiological signals, we should simply be more careful with them. So be much less public with those data, and only be public about sort of derivatives that we understand. So if it's physiological data from the heart, the pulse rate, for example, if we think that's okay to share, then we can do that, but we shouldn't share the raw signal. And the same with the EEG. We should maybe share some fun facts that we can derive from the EEG, but not the EEG signal itself. So that's the content of this paper that we wrote some years ago when we started doing these experiments, that we have to be super careful. And let me tell you that many of the big tech companies are very interested in this technology. So you probably already have heard about the efforts at Meta, Facebook, to build devices that can read your mind. And you've probably seen also Elon Musk's experiments on animals and so on. And so there are many also crazy visions for what this can be used for, and we should be careful. So super important that we keep it under democratic control and the control of the individual when we talk about data that can be so revealing as EEG signals. But yeah, super interesting discussion.
All right, thank you very much. I don't see any more questions, so that will be all. Thank you so much for the lecture. It was very interesting. Is there anything more you'd like to add?
Let me just thank you again for this opportunity, and say that it's really an invitation also. So let's hear about your ideas and so on. You're welcome to write me, of course, and start discussions about all this. So yeah, thank you again. Bye-bye.
Thank you so much.